Hello, everyone, and welcome to today's webinar, Increasing Efficiency in Clinical and Biotherapeutic Antibody Development. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Sartorius. To learn more, visit www.sartorius.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical issues, you can utilize this Q&A box and we will assist you directly. I'd like to now welcome our speaker, Daryl Cole, Application Scientist with Sartorius. Daryl, you may now begin your presentation.
Hello and welcome to this webinar. My name is Daryl Cole and I'm a scientist at Sartorius UK. Today I will be talking about increasing efficiency in clinical and biotherapeutic antibody development. So let's take a look at the agenda for today. We'll start the day with a discussion about antibody technology, what's currently available. Then we'll move on to hybridoma technology and the developments surrounding that particular type of technology. Then we'll move on to B-cell technology. And following that, we will have a look at the CellCelector Flex system, have a bit of an introduction to what its capabilities are. Then we will look at some of the proprietary products available for the system. So the nanowell technology will be the focus there. Then we will look at how Sartorius products can provide some solutions to a lot of the conventional challenges you'll have with antibody development. And finally, we will summarize and conclude from there. So let's take a look at antibody technology. The implementation of antibody technology for the development of therapeutics for a variety of diseases has increased exponentially, with more than 150 antibody drugs approved or in regulatory review since the first monoclonal antibody drug developed in 1986, an anti-CD3 orthoclone, OKT3. Antibody drugs have been developed for the treatment of various cancers, autoimmune disorders, viral infections, neurological and metabolic diseases. And some of the advantages to using antibody drugs are high specificity, the low levels of toxicity with few side effects, and good development success rates. As therapeutic antibody development has progressed, it has provided scope for antibody drug conjugates, bispecific antibodies, and also nanobodies as further developments of the therapeutic antibody. Monoclonal antibody research combined with other technologies has led to the development of chimeric antigen receptor T cells and CAR natural killer cells, CAR NKs. Due to their flexibility and high development success rates, monoclonal antibodies are a very sought after product in clinical and research fields. And therapeutic antibodies are also one of the most important research innovations in recent years for the treatment of disease, with different technologies that can be used for their production, which I'll go on to in the next couple of slides. Next, we'll take a look at one of the technologies used to develop antibodies from B cells, and that's hybridoma technology. In order to produce B cells that secrete the monoclonal antibodies desired by the researcher, typically an animal will be inoculated with a particular antigen, and then their B cells will produce monoclonal antibodies to target that antigen. These B cells are then harvested from the inoculated animal and characterized, and then the desired type is selected for development. Developments in the technology have established humanized monoclonal antibodies from target animals, which are far more useful in clinical settings, and these can be used in clinical settings for the treatment of disease and in research and development / drug discovery. So once you've isolated the B cell of choice that is producing the desired antibody, to generate large quantities of this antibody, the B cell needs to be immortalized. This is because B cells are very difficult to grow in vitro and they are quite short-lived. So you need some some method of immortalization in order to allow you to produce the required numbers of the antibody. So one of these ways is to use hybridoma technology. So what you do here is take a B cell and you fuse it with a myeloma cell, which is an immortalized cancer cell. And this fusing creates a hybridoma. The hybrid cell is able to replicate indefinitely, but it also produces the monoclonal antibodies that you're looking for. So it effectively produces infinite numbers of the target antibody. However, for all the benefits of this particular type of technology, there are drawbacks as well. And some of those are listed here. So the generation of hybridomas is very time consuming. It takes several days to weeks to produce a hybridoma. It's expensive as well. It requires multiple technological platforms and sophisticated procedures in order to successfully produce a fusion between a B cell and a melanoma cell. It's labor intensive. It's a very complex manual process and it requires highly skilled personnel, and it's highly inefficient. So for 5,000 B cells that you try to adapt into hybridomas, about one of those will survive the fusion and become a fully fledged hybridoma that produces the antibodies that you're looking for. On the next couple of slides, we'll have a little look at B cell technology. Similarly to how the B cells are harvested for hybridoma production, in order to collect B cells for other types of B cell technology, an animal such as a mouse is usually inoculated to start producing B cells that target the antigen of interest with their produced monoclonal antibodies. So again, the B cells are characterized and the desired type is selected for development. And as I said, it often follows a similar initial development process to hybridoma-based methods. However, it changes where, after characterization, the isolated B cells can be used to develop other methods of antibody generation, which are a little bit different to hybridoma technology. So as I previously alluded to, conventional cell culture-based methods aren't really sufficient for the growth of ex vivo B cells, as they're quite short-lived and difficult to maintain. They're not a very robust cell type outside of the body. So characterization of these cells and their products shortly after isolation is very important for developing other methods of antibody production before you lose the cells, they die or stop producing. Alternative methods of large-scale monoclonal antibody development include recombinant antibody production using host cell systems such as Chinese hamster ovary cells as a particular example. So CHO cells, or Chinese hamster ovary cells, are very susceptible to modification to produce monoclonal antibodies of interest in large quantities. The characteristics of CHO cells allow for post-translational modification of human biosimilars as well, so you can further modify your biosimilars and antibodies using this particular method. The cells are immortalized as well, and they have a highly homogeneous property in culture, so you'll have a very homogeneous population within your cell culture, which is what is ideal for antibody production. They're also very easy to grow in large-scale cell culture under defined conditions, such as in bioreactors. And on the right-hand side here, we have a nice image of the AMBER 15, which can be used for that particular purpose. And this allows the production of vast quantities of biologics. So you're growing your cells in a bioreactor and they're producing antibodies all the time, and then you can collect large quantities of those quite simply because the CHO cells are quite a robust cell line and they grow quickly and are very hardy. So now let's have a look at the CellCelector Flex, the cell picking system from Sartorius. So here is the CellCelector Flex system. So what we have on the left-hand side is the system itself. So you can see the automated microscope in the middle of the image with bright field, phase, contrast, and epifluorescence capability. On the left-hand side, we've got the robot arm. So you can see it's a precise robot arm for cell retrieval by aspiration, but also we can use a scraping tool for those adherent or hard to detach cell types. And then over on the right-hand side, this is how the whole system is powered. We've got the software and the joystick controller that allows us to analyze the images that the CellCelector produces, make our cell selections, document the selections that we've decided on, and also document before and after images so that you can see the results of your picking. And also this is where we control the robot's arm and all of the processes with the system. The CellCelector Flex has the capability for large application versatility, and this is due to the exchangeable modules. So if you look to the left-hand side, you can see the system, and circled in yellow is the module, the picking module that has been installed. The system will automatically detect which one that is, so you don't have to tell the system which module is installed. And there are 3 systems currently available. We have the single cell picking module, which uses glass capillaries, and that can pick up to a diameter of 220 microns. So that's good for your individual cells, small colonies, small organoids. And then we also have the adherent colony picking module, which uses reusable stainless steel tips. So you can see the tips there on the image in the middle. They can be autoclaved for reuse, and they have a range from 300 to 1,000 microns in terms of diameter. So you can pick larger colonies with those and larger organoids. And finally, we have the semi-solid media picking module, which is mainly used for picking from matrices such as Matrigel, and they use disposable plastic tips, and they range from 500 micron tips to 1200 micron tips. And they, as I say, they can be used for picking from semi-solid media, so spheroids and organoids mainly for that particular module. So now that we've taken a look in depth at the CellCelector Flex, let's take a look at the nanowell technology, which is very useful in cell line development and B-cell workflows. Here on the left is an image of a 24-well nanowell plate. So as you can see, it looks very much like a conventional 24-well plate. But if we zoom in to one of the wells, we can see that there are actually thousands of nanowells within. Because these plates have thousands of nanowells per well, it can provide a solution to conventional bottlenecks associated with B-cell screening and cell line development. There's up to 4,000 nanowells per single well in a 24 well plate format, which allows you to streamline your screening capacity because you can screen thousands of clones using the automated processes of the CellCelector Flex. The CellCelector Flex is so precise that it can pick individual cells or clones from within these nanowells inside these plates, something that you would never be able to do manually. And this can be done in an automated fashion as well. As we move on, we can have a look at some of the solutions to conventional challenges in antibody development using some of the systems produced by Sartorius. Some of the conventional challenges associated with hybridoma technology come in the form of significant bottlenecks, such as when isolating and characterizing B cells. Determining the best possible source cells for transformation into hybridomas is incredibly beneficial, but that can be quite difficult with conventional technology. It's also very important to increase the yield of your hybridoma while reducing the failure rate. So choosing B cells that are high producers but also are very robust will help greatly in this particular instance. In addition to all of this, the process of producing hybridomas is very time consuming and resource intensive. So automation of key processes would increase efficiency and save time and resources. So this is where the CellCelector Flex comes in with its automated technology. It allows for more precise characterization of cells, B cells specifically here, and at a much higher throughput than they would be through manual conventional techniques. You can also replace the labor-intensive, unreliable manual handling with a highly accurate, reproducible automated solution. And in addition to that, you can reduce time to results. So the CellCelector workflows can provide the opportunity to screen, characterize, and pick B cells within hours using a single system, as opposed to it taking days or weeks with conventional methods. And I'll go into this particular workflow on the next slide. Here I'll go over the B cell screening workflow to be used with the CellCelector Flex. So if we start here with the first part of the workflow, so that's the cell loading, it takes approximately 10 minutes. So this is the hand cell seeding of a single cell suspension into a CellCelector nanowell array. Following on from that particular part of the workflow, you would get to the single screening section, which is 15 minutes. It's an identification of single cell nanowells. So this would be performed on the CellCelector. You would scan and analyze your nanowell plate and identify your single cells. From there, you would then perform a secretion assay, which take between 2 and 3 hours. And using the CellCelector, you could automatically detect the secreting cells using a fluorescence-based secretion assay. I'll go into detail about these in the next slide. And then you would transfer your cells, so you'd export your particular hits to PCR plates for downstream processing. So you'd use the CellCelector to then go into those nanowells, pick the cells that you want, put them into your PCR tubes or plates, and then you could process those downstream. And finally, then you would produce a report. So the CellCelector would generate the required documentation of your particular experiment, and that would take approximately 15 minutes. So what you can see here is that the B cell screening and isolation workflow is a lot shorter, a lot faster than what you would see with conventional methods. On this slide, we can take a look at some examples of the in-well screening applications that can be performed using the CellCelector Flex. So the first one here is an assay with plate capture coating. So what's performed here is the antigen, protein A or protein G, is adhered to the nanowell surface. The plasma B cells are plated and allowed to settle into these individual nanowells. And once the plasma cell starts secreting its antibodies, these will adhere to the antigen that's coated on the plate, and then we can add a fluorescently labeled antibody to detect that through fluorescence. A second method is to use an antigen expressing reporter cell assay. So you'd use a reporter cell that has its own antigens bound to its surface. The plasma B cell will then release its secreted antibodies. They will bind to the reporter cell, and then you can use fluorescently labeled antibodies to bind to the secreted antibody on the reporter cell, again giving you a fluorescent signal, allowing you to identify your B cells that are producing your antibodies of choice. And finally, another option is to use the bead-based assay, where you'd use, in a similar way to the antigen-expressing reporter cells, you'd use beads instead. So these beads already have antigens coated on the surface, and it's the same kind of technique. So your B cell would secrete the antibodies of choice. They would bind to the antigen that you're hoping to bind to, that you're hoping to target, and then you would use fluorescently labeled antibodies to then bind to the secreted antibody to give you a nice fluorescent signal, so that then you can look at your nanowell plate and you can select those nanowells which have your B cells that are producing the antibody that you're looking for. So here we have an example of the nanowell B cell screening in action using one of the B-based assays in a nanowell plate. So for this particular example, 2 million B cells were obtained and 60,000 cells were loaded into one well of a 6 macro well nanowell plate to achieve an average occupancy of 1 cell per nanowell. The antigen used was EGFR, so this was what the antibodies were targeting. So if we see a signal that confirms that the antibodies being produced are targeting EGFR correctly. So on this first image here with the red fluorescence, this indicates a anti-human IgG conjugated Alexaflor 647, which binds to the antibody produced by the B cell. So where we see this signal is showing B cells that are secreting antibodies. This next image is in green, and this is Alexaflor 488, and this is for a Alexaflor 488 conjugated streptavidin, which will bind to the EGFR specific antibodies. So where we see a green signal, that's where those are the nanowells where we have antibodies being produced by the B cells, but they also bind to EGFR. And then on this next image, we have the overview of the two channels, so an overlay of images of the two channels where you can see green and red. So in the upper left nanowell that only has the red signal, that's producing antibodies, but they're not binding to EGFR. In the lower and in the two to the right, you've got antibodies that are being produced, and they're also binding to EGFR. So those two wells would be the two that we would want to isolate and pick using the CellCelector Flex. So on the right-hand side, on the left-hand side here, we have the the well in Brightfield prior to being picked, and then after picking, on the right-hand side, you can see we've removed everything from that well to then analyze further or use for downstream methods and techniques. On this slide is an example of how we can further characterize our B cells using some of the other systems in the Sartorius portfolio, such as the iQue shown here, and also we'll be looking at the Octet R8 on the next slide. So in this graph, what we can show here is the EGFR binding of our antibodies to CHO cells that are expressing EGFR on their surface. So the way this can be performed is by taking your CHO cells with the EGFR surface expression, combining them with your antibodies produced by your different clones, and then using a second antibody with a fluorophore to then bind to that antibody. Then you can wash, and then you can run your cells through the iQue, and you'll get signal depending on the binding level of the particular antibody that you've produced with your clones. So what we can see here is we have 3 clones that have very high binding, B06, E08, and G04. These have binding of over 100-fold higher compared to control. And then we also have some lower binding clones which have low to no binding of EGFR, these being B09, D08, and H01 as a few examples. We can then further characterize these clones' production using the Octet R8, which is an image of in the top right-hand corner. So where we saw samples in the iQue data, we can see that there's sample B06 and G04 here have a similar binding over the control when analyzed on iQue. But when we look at them using BLI, we can determine that B06 has lower affinity for EGFR than G04, a KD of 4.4 nanomolar versus 0.9 nanomolar. And the sensorgrams at the bottom here show further detail on the kinetics of binding for these two antibodies and also highlight a non-binding sample, H01, that was confirmed not to bind to EGFR by both iQue and Octet analysis. So the sample B06 displays a high OM rate, which you can see at in the middle graph here. This high OM rate is quite a steep curve, but also a high off rate. So there's a high dissociation as well, leading to a lower affinity than G04 on the left, which has a lower OM rate, but also a lower off rate, as you can see in the sensorgrams. So this data is useful because although the two antibodies bind to EGFR, their affinities impart different physiological properties. So B06 in the middle associates to EGFR quickly, but then dissociates quickly too, which provides a short-term effect, while G04 takes longer to bind, but dissociates much more slowly, providing a more stable, longer-term effect. So you can gather more information about each of these clones when you look at characterization on both the iQue and the Octet, in addition to what we've already looked at with the CellCelector. Another example of a nanowell-based assay for plasma cell characterization is highlighted in this schematic here, showing you the characterization but also isolation of antibodies from the bone marrow of a SIV-MAC239 infected rhesus macaque. So the schematic here on this slide shows a representation of a fluorescence-based platform where the nanowells are coated with streptavidin in yellow here at the bottom, and subsequently an SIV-MAC239 SOSIP664 trimer in blue is added, and that binds to the streptavidin. Then total bone marrow cells are added to the nanowells in a media containing April, IL-6, and a mouse anti-monkey IgG FITC, which is the green cartoon on the image there. And this binds to the antibodies secreted by the plasma cells. So on the left-hand side, we have a plasma cell that is secreting antibodies that are specific to the SIV-MAC. They're binding to that particular structure there, and then the anti-mouse, anti-monkey IgG FITC is then binding to those secreted antibodies and providing a fluorescent signal. On the right-hand side, the cell has been immobilized in the well, and it's secreting some non-specific IgG antibodies, so they aren't binding to the SIV-MAC239. The mouse anti-monkey IgG FITC is binding to these secreted antibodies, but they are just diffusing away, and so you're not seeing a fluorescent signal as you would on the left-hand side. So here's what you would see on the CellCelector in the software from the imaging. So on the left-hand side is bright field view of your cells and your nanowells. And then on the right-hand side is the fluorescent field. And the green cursor in the bottom is the diameter of the capillary that you'd use to pick. So what you can see on the left-hand side is a couple of cells in the nanowell. And then if you compare that to the image on the right, you can see there's a large fluorescent signal in the top in the top right, in the lower portion of that well, suggesting that this particular cell at the bottom is producing or secreting antibodies that are binding to the SIV-MAC. So these are producing the target antibodies of interest, And then again, in the lower images, you can see a similar trend there where we've got cells and they're secreting the antibody of choice, whereas there are some other cells within that same well and they aren't producing. So you can see the difference there in the signals that you get on the CellCelector. So it's quite clear and quite straightforward to see. And then you could just use the system to pick the cells that you would want based upon their secretion. So to conclude, based on what we've learnt today, we're able to use the CellCelector Flex to characterize and isolate B cells with desired phenotypes in hours with the speed and simplicity that the CellCelector Flex provides, as opposed to days or even weeks with manual or conventional methods of B cell characterization and isolation. The CellCelector Flex also has a full auditing process and documentation of your isolated cells, so you can get the source and destination location, and also it will provide before and after images so you can track your cells throughout the workflow, see where they've come from and where they end up. You also have the option of analyzing B-cell monoclonal antibody specificity, production and yield within the same well using a variety of nanowell-based secretion assays, which dramatically increases the productivity. It's also highly efficient using accurate automated protocols and removes the need for intensive, inconsistent manual handling of biological material, increasing reproducibility. And you can use just the one system for identification, isolation, and characterization of B cells. For further information, please take a close look at these web pages. The top one is an overview of the CellCelector Flex system. The middle one is an application note about B cell workflows and increasing their efficiency. And the bottom one is a CLD or cell line development application note utilizing the CellCelector. I look forward to answering any questions that you may have in the Q&A session after this webinar. Thank you for your attention today for this webinar. Please feel free to reach out to the email on the screen with any additional questions that you may have. Thank you.
Thank you, Daryl, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for today. Let's go ahead and get started. Our first question asks, what other applications can nanowell plates be used for?
So the nanowell plates can be used for a variety of processes, but CLD workflow optimization is a particularly efficient use of that technology, which reduces the resource requirements and time taken for limiting dilution experiments, as an example.
Wonderful, thank you. Our next question that we have here says, what magnification capability does the CellCelector have? For example, I would like to analyze very small structures such as exosomes.
So the CellCelector is available with objectives from 2 times all the way up to 40 times. So it should more than be able to cover your needs for exosome visualization.
Great, thank you, Daryl. And it looks like we have time for just one more question here today. That question says, some of these workflows take an hour or so to complete. How do you ensure the environment is conducive to cell health?
So the CellCelector, you can actually get a flow box to put the CellCelector in. So this is a solution to this potential challenge. So this is a custom laminar flow box for the system. So that provides heating, humidity, CO2 gassing functionality. So you can essentially have an incubator with your CellCelector inside. And so it can be performing experiments in ideal conditions, which is especially useful for delicate cells such as B cells.
Great. That sounds wonderful. All right. Thank you again, Daryl, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Sartorius, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.